Hello, grade 12 students. In this session, we're going to continue with our chapter about the immune system, about chapter 6, the role and components of the immune system. And here we move to document 4 about the cells of the immune system, page 121 on your book. So in this document 4, we are talking about the immune system. As you know that every system inside our body consists of organs and cells. These cells, they work together to defend the body against any invasion by microbes, bacteria, viruses, and others. So what are the cells of the immune system and what are their characteristics? So observation of a blood smear, it can be observed under the microscope. You have to the prepare the blood smear. You have to, for example, prick the tip of the finger by a sterilized lancet. So then place a drop of blood at 1 centimeter from the end of a slide, which is a glass, small glass. Then put another slide beside the drop of the blood. Then push the upper side rapidly toward the opposite end of the lower slide to form a thin layer of blood. You have to add a few drops of alcohol 95 degrees to fix the smear. Then wait for 2 to 3 minutes. Add a few drops of the iodine solution. Wait for 7 to 8 minutes. Rinse it with water, Distilled water. Then add a few drops of methylene blue. Then you have to wait for 4 to 5 minutes. Rinse the preparation with water and let it dry. Place the preparation on the stage of the microscope and observe it. Always, it will look like this. But if you wanna see the blood under the microscope, you have to stain it. So the staining will allow you to observe the stained blood smear like shown in document 8. Different leukocyte population. هيدا اللي بتهمني. Observation of the blood smear, it's for your information, general information. So paragraph 2 talking about the different leukocyte population. We have different leukocyte population that one can observe either circulating into the blood, بسموهم circulating leukocyte, or present in the body organ, بسموهم tissue leukocytes. All leukocytes, they are derived from the same progenitor. From the same progenitor, which means that they are of the same source, as shown in document B. Origin of leukocytes, in the bone marrow, we have something called pluripotent stem cell. Pluripotent stem cell, it can be differentiated in a different type of cell, either lymphoid stem cell or myeloid or lymphoid, that we talked about them in the previous session. Myeloid, leukocytes that derive from the myeloid, they are the granulocytes, the monocytes, and the mast cells. Here about the granulocytes, in that they have a diameter, they have a multilobed nucleus first, and they have granules in their cytoplasm, and they are of three types: eosinophils, basophils, and neutrophils. Granulocytes, they have a diameter of 10 to 12 micrometers. They are shown in document C. These are the granulocytes population: neutrophils, eosinophils, and the basophils. So here, the granulocytes, they represent about 67% of the whole leukocyte population. They are produced in large quantities and they live for 2 to 3 days. Monocytes, حكينا عنهم, they are found in document D also. These are the monocytes. They have a role in phagocytosis. So monocytes, their diameter is 10 to 12, 18 micrometer, and they have a horseshoe-shaped nucleus. They represent about 6% of the total leukocytes, and they migrate into the tissue where they become macrophages. What do? They do the process of phagocytosis. They can live up to several days or even several years. Mast cells, حكينا عنهم, mast cells, they have a role in allergy. They are leukocytes having ground in the nucleus and a cytoplasm that is rich in granules filled with histamine. They possess receptors of certain antibodies and they found in the connective tissue and in the mucosa. This is about the myeloid. So myeloid in three types: granulocytes, monocytes, and mast cells. However, the lymphoid cells, leukocytes that derive from the lymphoid stem cells, they are either the B or the T lymphocytes. B and T lymphocytes that are found in document E. This document E is showing the B and T lymphocyte. This is B lymphocytes. This is a plasma cell. We're going to know a difference between that. And this one in paragraph 3, this is the T lymphocyte in figure 3. So here, what do we want from this step by step? So our both lymphocytes, they are characterized by being smooth with very large nucleus occupying all the cytoplasm, have a large nucleus, and they have a small cytoplasm. And here we don't have to study the details at there, how many micrometers and so on. They represent 25% of the total leukocyte population. B and T, they are characterized by their receptors. شو عندهم البي وشو عندهم التي؟ B lymphocytes, they are characterized by the membrane receptors. They have antibodies. So whenever you see a large rounded nucleus, a small cytoplasm, this is a lymphocyte. What determines whether it's B or T? It's the receptors. If it has antibodies, so it's B lymphocytes. However, if it's T lymphocytes, it has special type of receptors that bind to antigens. They are called the T cell receptors, the TCRs. Upon their activation, B lymphocytes, they will differentiate into plasma cells. Plasma cells, they are able to secrete the antibodies. In the plasmic reticulum, you hear about this ER, endoplasmic reticulum. The endoplasmic reticulum has an important role in the production of the proteins, and one of these proteins is the antibodies. So whenever we move from a lymphocyte, from a B lymphocyte toward a plasmocyte, which is a mature B lymphocyte, or a plasma cell, which is a mature B lymphocyte, the number of endoplasmic reticulum of ER will increase. Why the number of ER will increase? ER is the site of production of proteins. So number of sites of production of protein will increase in the plasma cell. Why? Because ER produces proteins. So it produces a type of protein which are the antibodies. So antibodies, they are a special type of proteins. So whenever they are produced, they need more and more endoplasmic reticulum. They need more ER. However, we have something called the mitochondria. And if you know the mitochondria, this is the source of energy. So number of mitochondria in the plasma cell will increase since it's the source of energy needed for the production and for the release of antibodies. Upon activation, as I said, B lymphocytes, they differentiate into plasma cells that secrete antibodies. These antibodies, they circulate in the plasma. However, T lymphocytes, they include two groups, and pay attention to this. Here, T lymphocytes, حكينا عنهم yesterday. We said that T lymphocytes, they include T helper and T cytotoxic, or the T killer. T helper, بيعطوها symbol T helper, T capital and small h. Not a small h, but it's capital H, but written in a small transcript. So here, TH, they are the T helpers. These T helpers, they will help other cells involved in immunity. They have something called a CD4. This molecule called CD4, it has an important role. We're going to discover it in the coming sessions. But CD4 موجودة على T helper. So T helper has a molecule is called CD4. That's why these T helpers بيسموهم T4 lymphocytes. Why T4? Because they have the CD4. So they are called T4 cells. So from now on, whenever we talk about T4 cells, so we are talking about T helper. Whenever we talk about T4, we are talking about T helper. Why? Because it has CD4, so سموها T4 cell. However, the other type of cell is called the cytotoxic T cell. Cytotoxic T lymphocytes, they give it T and C here. So T cytotoxic or cytotoxic T lymphocytes. These T cytotoxic mean that they are toxic. They kill the body cells infected by microorganisms. They have a special molecule, a small CD8. This CD8 is found on the surface of T cytotoxic. That's why بيسموا T cytotoxic بيسموهم T8 cell. Why? Because عندنا هيدا المولكيول اللي هو CD8. So T helper بيسموهم T4 because they have CD4, and T cytotoxic or T killers بيسموهم T8 because they have the CD8 molecule. So the first part, they need observe a stained blood smear under the microscope. They may label drawing of what you see. I don't want this. Moving to part two. Neutrophils, eosinophils, and basophils, they are termed granulocytes, or they are called polynuclears. So as I said, neutrophils, eosinophils, basophils, they are types of granulocytes, where the types of granule, whether acidic, basic, or neutral, it differs. So here, granulocytes, so here because they have granules, and polynuclears because they have a multi-lobed nucleus. That's why we call them polynucleus. Here we have to talk about the type of the characteristic that allow the cell 2 to accomplish its function. Cell 2 where? Cell 2, which is cell 2 in document E. How are the characteristics, or what are the characteristics? I said that we have increase in the number of endoplasmic reticulum, increase in the ER, and we have increase in the Golgi apparatus, okay, or the mitochondria also. So mainly the mitochondria, which is the source of energy. So I don't want all of them. I need only some of these parts of the following documents. So I need part 2 and part 4. So what granulocytes, they are called granulocytes, where they are called granulocytes because they contain granules. Why they are called polynucleus? Because they have multi-lobed nucleus. A cytoplasm of B cell enlarges the number of ER. So the number of endoplasmic reticulum will increase, and this is the site of production of proteins. And we have the number of mitochondria will increase. So this is the site of where the energy is produced. So here we finish document 4 talking about the types of the immune cells. تعرفنا على البي lymphocyte, T lymphocyte, تعرفنا على granulocytes and others. Okay? Moving now to document 5. Lymphoid organs, page 123. Lymphoid organs, in addition to the cell, immune system includes organs. These organs, they are the sites of production and maturation of the lymphocytes and where the reaction will be triggered. What are these lymphoid organs? We have the primary and we have the secondary. So primary lymphoid organs. Primary lymphoid organs, already we talk about them in the introduction. We talk about the bone marrow and the thymus. So in order to determine the site of production and the maturation of lymphocytes, experiments were carried on 3 lots of mice. These experiments are realized and the results obtained are illustrated in document A. Note that irradiation, and I told you this before, irradiation is a treatment that involves the destruction of the dividing cell and therefore destruction of the bone marrow. Yani irradiation with X-ray, it destroys the bone marrow. Already we have it in the quiz, and already I mentioned it in one of the live sessions. So we have here 3 lots of mice, lot A, B, and C. In lot A, we have irradiation plus a graft of bone marrow. So we destroy the bone marrow, then we grafted a bone marrow. Results, production of B and T lymphocytes. However, in lot B, we have ablation of the thymus. We remove the thymus. Then we did irradiation. Then we grafted the bone marrow. So ablation of the thymus, شلنا thymus. We did irradiation, destruction of bone marrow. Then we did grafting of bone marrow. So we have only bone marrow. What happens? Production of immature T and mature B. So thymus is not found. So the thymus is responsible for the maturation of the T lymphocytes. However, in lot C, we did ablation of the thymus plus irradiation plus grafting of the thymus. So we did ablation of the thymus, so we removed the thymus. Then we did irradiation by X-ray, so we destroyed the bone marrow. Then we grafted thymus. So we have only thymus. We don't have bone marrow. So there is no production of B and T. ما في عندي لا B ولا T. So this means that the bone marrow is responsible for the production of B and T. Even though we have the thymus, thymus has no role unless it matures the T. So where is the T? T is produced in the bone marrow. إذا ما كان عندي bone marrow يعني ما في عندي لا B ولا T. Lymphocytes maturation. Maturation is the genetic mechanism by which lymphocytes they become immunocompetent, meaning that become functional, they can work, and they are able to bind to specifically non-self antigen. نحن بنعرف نحن immune system it attacks the non-self. هلا بعد شوي حنحكي about some types of cells that attack the self. They attack the cells, and this will cause a special type of disease. Is called autoimmune disease, where the problem is autoimmune disease, where the immune system attacks its own cells. بيهجم على السيلز تبعوله بنسميهم autoimmune disease. هنحكي عنه. So during maturation, prior to any non-self antigen encounter, قبل ما نشوف any non-self, specific membrane receptors are formed, and each lymphocytes acquired one variety of the membrane receptors among a virtually unlimited number of possible varieties. This receptor diversity constitutes the receptor reservoir of the individual. So here, the lymphocytes are then screened in the bone marrow. B lymphocytes, that's why they are named B because of the bone marrow. They bind to self antigens. Whenever they bind to self antigens, they are eliminated. يعني البي lymphocytes whenever they bind to the self antigen دغري my body will cause their elimination. دغري بيقتلهم وبيتخلص منهم. This because they will kill my cells. So whenever they bind to any self antigen, they will be eliminated here. And whereas the others are preserved, others that do not recognize or that do not move to the self antigens, they are preserved and will keep in working. Okay, so here let's continue with the uh, thymus. A double selection takes place. A double process selection takes place. Why? Because we have in the thymus a double selection will take place for the T lymphocytes. Hence the name T because they are in the thymus. T lymphocytes and the double selection. Whereas B lymphocytes can عندها بس if إذا بتتعرف على السلف antigen will be eliminated. إذا ما تعرفت على السلف antigen it will not be eliminated حتكفي their normal life. يعني نحن البي lymphocytes مش لازم تتعرف على السلف antigen. إذا تعرفت على السلف antigen يعني عم تقتل لي my cells. So here it's a one type selection. While in the thymus and the double selection process takes place for T lymphocytes. Hence their name T thymus. T lymphocytes they bind first to HLA. So here T lymphocytes or receptors can bind to HLA of the self. They are preserved. يعني بيتعرف على HLA. إذا تعرفوا على HLA they will be preserved. Others they will be eliminated إذا ما تعرفوا. The lymphocytes whose receptor can recognize the self peptides are eliminated while others are preserved. كل الحكي اللي حكيته هنتفرج عليه بهيدي document B lymphocytes maturation. We have an immature lymphocyte gives rise to several lymphocytes, each having a different specificity. Elimination of the autoreactive lymphocytes. And all lymphocytes they bind to the self antigen. These two, the green والred, and here is the purple and إذا بدك مثلا الred الثانية, so they bind to the self antigen. So what they do, they are eliminated. So here they are eliminated, and only these three they are preserved or they persist. That's why I'm going to illustrate this drawing a diagram illustrating the maturation of B and T lymphocytes. We're going to move in this. In the bone marrow, أنا هستعمل legend. This arrow, the straight arrow, is recognized. For example, the curvy one, it does not recognize, and this arrow means results. So let's start first. This is a concept map. Sure, you have to draw it on one page, but here the one slide doesn't fit. So you have to draw it using a pencil, give it a legend, a title, and sure should be drawn using a pencil. So here, when the B cells, they recognize the self antigen. This one is recognized, and this does not recognize the curvy one. So when the B cells recognize the self antigen, تعرفوا على السلف antigen يعني حيقتلوا للسلف antigen. So these B cells will be dead. هتموت هيدي البي cells هيقتلوها. However, when the B cells do not recognize the self antigen ما تعرفوا على self antigen, so these B cells will survive. However, the T cells اتفقنا عندي double selection process. Here the first selection and here the second selection. First, they have to determine to recognize the self antigen. تعرفوا على self HLA, they will survive. ما تعرفوا على self HLA, they will die. So here إذا تعرفوا على self HLA قلنا they will survive. إذا ما تعرفوا they will die. إذا تعرفوا على HLA خلصوا أول process. ثاني process they have to recognize. If they recognize the self antigen they will die. يعني حنرجع نفس السيل تاعون البي نفس الستep. But إذا ما تعرفوا على self antigen they will survive and they will continue their life. So البي lymphocytes ممنوع يتعرفوا على self antigen. إذا تعرفوا على self antigen they will die, and the B cells should not recognize the self antigen. That's why they will survive. T cells عندنا double selection process. First process they have to recognize the self HLA in order to survive. إذا ما تعرفوا على self HLA يعني هن ما تعرفوا على السيلز تبعولي, these cells will die. تعرفوا على self HLA خلصوا first process. Moving to the second process, they have to recognize the self antigen, so they will die. إذا ما تعرفوا على self antigen, they will survive. So إذا لاحظتوا هيدي common بيناتهم. Second process is the same in both of them, in the B and in the T. But بالتي عندي double selection process in the HLA لازم يتعرفوا على self HLA. So here, this is the description of the first paragraph. Moving now to the secondary or the secondary lymphoid organs. حكينا about the secondary lymphoid organs, حكينا about the spleen and the lymph node. So when they have undergone maturation, lymphocytes, they leave the primary lymphoid organs and they migrate toward the secondary lymphoid organs, mainly to the lymph nodes and the spleen. This is the spleen, حكينا عنه the spleen, حكينا about the lymph node. The lymph node, they are small organs lining up like a string of beads along a network of vessels. They are called lymphatics. The lymphatic network collects extracellular fluid from the tissues and brings it back to the blood vessels. This fluid شو بنسميه is called the lymph. Okay, this fluid is called the lymph. The lymph nodes, they are the site of responses against antigens drained by the lymph from infected tissues. However, the spleen is located in the upper left part of the abdomen at the site of reaction against infections or antigens carried out in the blood. فنحن حكينا spleen against blood infection, lymph node against tissue infection. So the lymphocytes, they will circulate continuously through the lymph and the bloodstream between the different secondary organs in order to maintain permanent surveillance of the external body against foreign elements. So هيدي هي البروسيس اللي عم بتصير نحن at the level of the spleen and at the level of the lymph nodes. So here, based on the results presented in document A, what can you deduce? This is document A. Document A اللي كان بيحكينا about the experiment, irradiation, the grafting of bone marrow, and so on. Bس we will remove, let's talk about the lymphoid organs. Already حكيناهم بالintroduction. بس حكينا about primary as the site of production and maturation of leukocytes. These are the bone marrow and the thymus. However, the secondary lymphoid organs, they are the site of functioning of leukocytes where the immune reaction took place. They are the spleen and the lymph nodes. So هون أنا حأعمل for example مثل عم بعمل interpret، ما عملت مثل ما هو بده بالسؤال. هو بده based what can you deduce، بس بده deduction. So here in mice of blood A that have bone marrow and thymus, there is production of B and T lymphocytes. So هيدي control lot. A control experiment used to compare and explain the different experimental results. Mice of blood B, what do we have? They have bone marrow but no thymus. So we have production of mature B but immature T. So this indicates that the bone marrow matures the B, but the thymus is responsible for the maturation of T lymphocytes. In mice of blood C that have thymus but not bone marrow, there is no production of mature B or T. This means that the bone marrow is the site of production of both of them, B and T. So deduction, B and T, they are produced in the bone marrow. Bone marrow matures only the B, whereas thymus matures the T lymphocytes. Part 3. They said part 2 already, we drew the diagram. We're going to talk about this. Why? Or the lymphocytes whose receptors bind to self-antigens, they are eliminated. Lymphocytes, whenever they bind to self-antigen, they are eliminated. Why? Because they will cause an autoimmune disease. يعطيني شيء اسمه autoimmune disease. Part 3, they said, part 3 before part 4. Part 3, they said nude mice are mutant mice. That whenever you hear about nude mice, they are mice that are deprived of thymus. What does this deficit impact their different leukocyte population? So part 3 I'm بقول nude mice. Nude mice from now on, whenever you hear the word nude mice, يعني هن mice من أول ما خلقوا they have no T lymphocytes. So they have no thymus, sorry. So no thymus, they have no mature T lymphocytes. Okay? So part 3, so all the number of white blood cells, they are normal except for the T cells which are immature. So part 4 اللي حكينا عنه about the autoimmune disease. Why are the lymphocytes whose receptors bind self-antigens, they are eliminated during maturation. يعني كل lymphocyte تتعرف على السلف انتيجين بينعمل لها elimination. Why? Because إذا تعرفت على السلف antigen, it will lead to the killing or the death of my cells. So this will lead to something called autoimmune disease. تعطيني شيء اسمه autoimmune disease. This is part 4. Since the lymphocyte that can recognize the self then can cause autoimmune disease. في كتير ناس عندهم إياه هذا autoimmune disease. Okay? This is part 2, the figure that we have to draw it regarding the bone marrow and its function and recognition. So next time, we're going to start with document 6. We have to talk about document 6, antigen recognition by B lymphocytes. And we're going to move to document 7, antigen recognition by the T lymphocytes as well. And then we can move to the exercises of the chapter.